Usually when we think about a multi-million-dollar product, say a hypercar or some piece of advanced aerospace technology.
Right, you picture this pristine, perfectly engineered environment.
Exactly. You picture a clean room from a sci-fi movie, massive permanent structures, lasers, titanium.
Absolute engineering rigidity.
Yeah, we instinctively associate permanence with high value.
It's a very natural human assumption. I mean, we expect the tools making the most expensive, complex things on Earth to be just as imposing and, permanent as the products themselves.
But then you step into the world of modern biomanufacturing.
Yeah.
And suddenly that whole sci-fi factory image is just completely turned on its head.
Oh, completely.
We are looking at a manufacturing landscape where the absolute most advanced life-saving medicines on the planet are being produced inside what are essentially highly engineered disposable plastic bags.
And that single shift, moving from rigid steel to flexible plastic, that is the absolute definition of an industrial paradigm shift.
It's wild.
It completely rewrites how we understand the economics of healthcare, capital expenditure, and even global supply chains.
Well, welcome to the deep dive. Today, we are taking you on a journey into the structural economics, the intense history, and the 10-year trajectory of the bioprocessing industry.
Right.
And we are anchoring this entire analysis on one of the most fascinating companies you might never have actually heard of, which is the German manufacturing giant, Sartorius.
Our mission today is to unpack how a company like Sartorius has positioned itself at the absolute center of this global shift in how medicines are made.
Because it's not an accident.
No, not at all. We need to look at the inflection points that built their empire, the underlying mechanisms that keep their customers completely locked in, and the structural forces that are currently threatening to disrupt their dominance.
And to do that, we have an incredible stack of sources to work through today. On one hand, we have a really rich mix of Sartorius's own financial reports and corporate presentations.
Stretching all the way from 2017 through to their most recent 2025 annual report.
Right. And that includes their Q2 2025 earnings call.
But on the other hand, we have something much more candid, which I think is where the real story is.
Oh, absolutely.
Right.
The expert network interviews.
Exactly. We are contrasting management's highly polished, forward-looking narrative with these raw, unfiltered transcripts from expert network calls.
These are off-the-record conversations with former industry executives, customers, consultants, and direct competitors.
It gives you a view right behind the curtain. It highlights the price friction, the competitive anxieties, all the stuff you just don't get from a glossy annual report.
So why should you care about bioprocessing? I mean, think about the medications in your own cabinet or the breakthrough treatments you read about in the news.
Biologics.
Yeah. Behind every miracle biologic drug, every breakthrough gene therapy, every mRNA vaccine.
Medicines that can cost up to $2.1 million for a single dose, like Novartis's Zolgensma.
Exactly. Behind all of that, there's a highly lucrative, heavily concentrated supply chain holding it all together.
So this isn't just a story about healthcare.
No, it's a masterclass in building an impenetrable economic moat.
And to truly understand the power of that moat, we have to understand the castle they built it around, right? Because the biomanufacturing industry didn't start with flexible plastic.
Far from it. We have to look back at the formation of this industry and the massive structural constraints of what we call the stainless steel era.
Right. Let's unpack the mechanics of that era. When we talk about the roots of biomanufacturing, we are talking about an industry defined by towering, rigid stainless steel bioreactors.
We're talking 20,000 liter to 80,000 liter vats. Just massive.
And some legacy facilities built by massive contract development and manufacturing organizations or CDMOs like Samsung Biologics or Lonza, they housed hundreds of thousands of liters of capacity in these colossal steel tanks.
And from what I understand reading the sources, the structural forces of that era dictated everything about the business model.
Oh, absolutely. The initial capital outlay was astronomical. I mean, you don't just buy a 20,000-liter steel tank.
Right. You have to build a whole building around it.
You build an entire architectural footprint. You build massive subfloors for weight bearing. You install miles of permanent rigid piping to move the fluids in and out.
And the water systems, right?
Yes, you have to build massive water purification plants just to support the facility.
But the crazy part, at least to me, is that the real structural bottleneck wasn't even building the facility. It was actually running it.
The operations were a nightmare.
Because in a stainless steel facility, the production lines are rigid. When you are making a biologic drug, you're using genetically modified living cells as microscopic factories to secrete the medicine.
It's essentially a brewing process.
Right. And a single rogue protein, a single stray bacterium left over in that tank, could ruin an entire multi-million-dollar batch.
The risk of cross-contamination is catastrophic. So once a batch of a biological drug is finished, you can't just pour the next batch into the vat.
You have to clean it. And not just, like, rinse it out.
No, you undergo massive, highly regulated cleaning and validation efforts. We are talking about clean-in-place and steam-in-place protocols.
Basically boiling the tank.
You are essentially boiling the tank and the miles of rigid steel piping with caustic chemicals and highly purified steam for days.
Which means the facility isn't producing anything during that time.
Exactly. The downtime is huge. The cleaning effort requires massive consumption of water and energy. And time is money when you have a facility that costs hundreds of millions of dollars to build.
You need that facility running 24/7, but you are constantly pausing to sterilize it.
Which perfectly sets the stage for the first massive inflection point in this industry.
Technological disruption, or what the industry calls process intensification.
The pivot to plastic bags.
But delving into the science here reveals something really crucial, which I didn't realize until I read the sources. The flexible plastic bags didn't magically force the industry to change on their own.
No, the technology shifted because the biology finally allowed it to.
Right. The industry developed what the sources call high-yielding clones.
And that biological leap is the catalyst for everything that follows. In the early days, those genetically engineered cells were, frankly, relatively inefficient.
They didn't produce a lot of the actual drug.
Exactly. They didn't produce a high concentration of the desired drug, say a monoclonal antibody per liter of fluid. So you literally needed a 20,000-liter vat just to harvest enough of the final drug to make the batch economically viable.
But as genetic engineering improved, scientists created cell lines that were absolute powerhouses. The yield per liter just skyrocketed.
It's an exponential gain in efficiency.
So the output you used to get from a massive 20,000-liter legacy stainless steel tank could now be produced in a 2,000-liter bioreactor.
And the moment the required volume drops to 2,000 liters, the physics of the manufacturing floor completely change.
Because it's smaller.
Right. You don't need a massive steel tank capable of withstanding the hydrostatic pressure of 20,000 liters of fluid. You can suddenly use a pre-sterilized single-use plastic bag.
Supported by a much smaller, flexible framework.
The operational efficiency gained here is just staggering. When you are done with the batch, you don't spend a week boiling the tank with chemicals and steam.
You literally just take the plastic bag out and dispose of it.
You snap a new, gamma-irradiated, pre-sterilized bag into the frame, and you start the next batch immediately.
The downtime drops to almost zero.
And the water and energy consumption plummets. Plus, the risk of cross-contamination is virtually eliminated because the product only ever touches the single-use plastic, which is immediately discarded.
But the economic mechanism here is what really stood out to me. This biological and technological leap didn't just change how fast companies could make drugs.
It completely lowered the barrier to entry for who could make drugs.
Exactly. Consider the risk profile of drug development outlined in the Sartorius presentations. The probability of a drug candidate advancing from clinical phase I all the way to final regulatory approval is, what, roughly 10%?
Roughly 10%, yes.
So 90% of the time, the drug completely fails in trials. All that research, all that time, it just vanishes.
Right. And in the stainless steel era, if you were a small biotech startup with a promising molecule, you faced an impossible capital dilemma.
Because you couldn't afford a steel factory.
You couldn't afford to build a massive permanent steel factory for a drug that had a 90% chance of failing in clinical trials. The capital risk was simply too high.
So you were entirely dependent on outsourcing.
Yes, outsourcing to those massive CDMOs waiting in line for their massive tanks.
But with single-use technology, that capital risk totally evaporates. You don't have to build permanent infrastructure.
You lease a modest lab space, and you buy flexible, scalable, single-use systems from a company like Sartorius.
And if the drug fails?
If the drug fails, you haven't stranded hundreds of millions of dollars in permanent steel. You just stop buying the bags.
This specific inflection point created very clear winners and losers.
Very clear. The winners were the single-use innovators providing the exact picks and shovels needed for this new, flexible gold rush.
Companies like Sartorius and Thermo Fisher captured massive market share. The data in our sources shows Sartorius saw their sales revenue grow at an incredible 16% compound annual growth rate from 2013 to 2023.
16% CAGR over a decade in manufacturing is phenomenal. And the losers were the inflexible legacy players.
The massive CDMOs wedded entirely to legacy steel. They were forced into an incredibly painful, capital-intensive transition.
One of our expert sources from the network transcripts explicitly noted this. They said that facilities stuck with traditional 40,000-liter setups are essentially trapped producing legacy molecules that were developed 5 or 6 years ago.
Because they can't easily switch to the new high-yielding clones.
Right, because changing the biological clone means changing the entire manufacturing process.
And changing the process requires going back to the FDA or EMA. You have to refile regulatory paperwork and potentially run entirely new clinical safety trials just to prove the new clone produces the exact same drug as the old clone.
It's a massive technological lock-in. If you started in steel, you're pretty much trapped in steel until the drug goes off patent.
But if you are a new drug entering the pipeline today, the data suggests you are almost certainly starting in single use.
Almost without exception.
Which brings us to the turbulence of the last few years. The second major inflection point we need to talk about. The violent demand shocks and the brutal reality of the capital cycle.
Right, because the bioprocessing industry experienced an unprecedented roller coaster starting with the COVID-19 pandemic.
Suddenly, the entire world needed vaccines and therapeutics, and they needed them immediately.
This demand shock forced a massive, rapid adoption of single-use technology. Global manufacturing capacity expanded at a breakneck pace. Customers were basically buying every bioreactor, every filter, every bag they could get their hands on, regardless of price.
It was panic buying, an influx of capital and urgency that warped the normal growth curve.
But as supply chains normalized, the industry entered a severe destocking phase.
Because customers had hoarded massive stockpiles of these plastic bags and filters during the pandemic out of pure panic.
Exactly. And once the panic subsided, they simply stopped ordering. They spent quarters just burning through the inventory sitting in their own warehouses.
And right alongside that destocking phenomenon, the wider macroeconomic environment shifted violently. Interest rates went up.
And the venture capital money that historically fuels the early-stage biotech startups, the very top of the drug development funnel, started to dry up significantly.
We also saw a painful contraction in some of the most hyped advanced therapies. The expert sources pointed out that gene therapies, like that $2.1 million Zolgensma treatment we mentioned earlier, saw a major contraction.
And not because the science failed.
Right. It was because of the sheer logistical hurdles in getting insurance companies and national healthcare systems to actually figure out how to pay for a multi-million-dollar single-dose cure.
The reimbursement friction was real. So combining destocking, a venture capital funding crunch, and reimbursement friction, the industry faced significant headwinds leading into 2024 and 2025.
Okay, but I have to aggressively push back on management's spin regarding this exact point.
Go for it.
Because if you read the Q2 2025 earnings call transcript, Sartorius management claims they are largely insulated from this early-stage biotech funding crunch.
Yeah, their specific defense is that roughly 60% of their bioprocess solutions revenue comes from commercial manufacturing drugs that are already approved and being sold.
And if you add in late-stage phase 2 and phase 3 trials, they claim that number jumps to well above 80%.
Management is arguing that their revenue is sticky. It's highly concentrated at the end of the pipeline where the drugs are already successful, funded, and being manufactured at scale.
They view this as a protective shield against venture capital volatility.
They do.
But isn't that just whistling past the graveyard? I mean, logically, the biopharma pipeline is a multi-year conveyor belt.
It is.
If the top of the funnel, the early R&D and phase I trials, dries up today because the venture capital is gone, doesn't that eventually starve the commercial stage in 3, 5, or 7 years? You cannot have newly approved commercial drugs driving revenue in 2030 if you don't fund the early research in 2025.
That delayed causality you are pointing out is a highly critical structural vulnerability that management often glosses over in quarterly calls.
They just focus on the immediate numbers.
Right. Management is hyper-focused on the immediate quarter-to-quarter stickiness. The validation lock-in does protect their commercial revenue in the short to medium term.
But the math of the pipeline is inescapable.
You can't cheat the conveyor belt.
If you stop putting raw materials, new biotech startups onto the belt today, there will inevitably be a gap in commercial approvals down the line.
So the 80% insulation claim is basically a snapshot of today's health, not a guarantee for the industry's health in the 2030s. It is entirely a lagging indicator. However, to provide the full context from the experts, the broader macroeconomic fundamentals provide a very high floor for the industry, even if the early pipeline thins out temporarily.
Like aging populations.
Aging populations globally, the massive and growing volume of legacy drugs still being produced, and the rapid rise of biosimilars, which are essentially generic versions of biologic drugs.
So those ensure that established commercial manufacturing will continue running at high volumes.
Exactly, keeping the lights on and the margins healthy for suppliers like Sartorius.
This friction between the capital cycle and the underlying demand naturally shifts our focus to the profit pools themselves.
Where the actual money is made.
We understand how the industry moved to flexible single-use tech, and we see the turbulence of the recent destocking phase. But the most fascinating part of these sources is how that shift completely rewrote the profit pools.
To map the profit pools, we first have to characterize the market structure. The bioprocessing supply landscape is not a highly fragmented market with hundreds of tiny players fighting for regional dominance.
Now, it is a highly concentrated oligopoly at the top.
It's dominated by the big four. You have Danaher, operating mostly through their massive Sativa brand. You have Thermo Fisher, you have Merck KGaA, operating their Millipore Sigma unit, and you have Sartorius.
The reason this market condensed into an oligopoly is because the barriers to entry are astonishingly high. And the foundation of those barriers brings us back to the regulatory environment.
This is the ultimate economic moat.
When I was rolling through the sources, the economic mechanism of this moat just blew my mind. Let me offer an analogy to ground this.
Let's hear it.
Think about buying a specialized home printer. We all understand the classic razor and razor blade business model. The manufacturer sells you the printer at cost, or even at a loss.
Right, and then they make all their profit gouging you on the proprietary ink cartridges over the life of the machine.
A classic, highly effective model of value capture. But in the bioprocessing industry, it is that exact model on steroids because it's enforced by the federal government.
That's the kicker.
It's not just that Sartorius sells a pharmaceutical company a bioreactor frame and simply hopes the customer continues buying Sartorius plastic bags and filters.
Yeah.
It's that the FDA legally mandates that the customer must use that exact bag and that exact filter for the entire lifespan of the drug.
The regulatory mechanism here is profound. When a pharmaceutical company gets a biologic drug approved by the FDA or the EMA, they aren't just getting the chemical or biological formula approved.
They are getting the exact step-by-step manufacturing process validated.
The process is the product.
So following the analogy, it's as if the government legally mandated that you must use HP ink in your HP printer. And if you ever want to switch to a cheaper generic ink from a new supplier, you can't just swap the cartridge.
No, you have to spend millions of dollars and potentially years running new safety trials to prove to the government that the generic ink didn't fundamentally alter the final document.
The switching costs are effectively punitive.
Once a Sartorius product, be it a specialized virus filter, a mixing bag, or even a piece of proprietary tubing is specified and written into a customer's validated regulatory filing, it is locked in.
The friction required to switch suppliers is so high, and the risk of delaying commercial production during revalidation is so massive that customers almost never change suppliers unless a catastrophic failure forces them to.
And that regulatory lock-in translates directly into legendary revenue stickiness. According to the 2024 investor presentation metrics in our stack, roughly 80% of Sartorius's bioprocess solutions business is recurring revenue from consumables.
They sell the hardware once, and they sell the highly profitable, validated consumable bags and filters for the 10 to 15-year commercial lifespan of the drug.
But a deeper analysis of the expert network transcripts reveals that not all recurring revenue is created equal. The profit pools within that 80% consumable bucket are actively migrating.
And to understand why, we have to clearly differentiate between upstream and downstream processing. I found this distinction critical in the sources.
It's the most important division on the factory floor.
Upstream processing is essentially the cultivation phase. It's the actual biological production of the drug. It involves the cell culture media, the liquid nutrients, and the cells themselves sitting in the bioreactor, multiplying and secreting the target protein.
Exactly. It is the messy biological brewing phase.
And downstream processing is everything that happens after you harvest that brew.
It is taking that highly complex biological soup and isolating, purifying, and filtering out the exact desired drug molecule from the surrounding cellular debris.
Downstream is the separation, the chromatography, the virus clearance,
Hmm?
and what the unfiltered expert interviews reveal is a massive push by pharmaceutical customers to commoditize the upstream products.
Basic culture media, simple buffer solutions, standard mixing bags, these are becoming easier for competitors to replicate.
The customers are actively organizing to break the supplier lock-in for these simpler upstream products. The sources explicitly mention the BPOG initiative.
Right, the Bioforum Operations Group.
From my reading, this is a powerful consortium of biopharmaceutical manufacturers. They are banding together to push for standardized testing protocols across the industry.
Specifically, they want universal data on things like extractables and leachables.
What does that mean in plain English?
In plain terms, they want standardized safety data on what microscopic chemicals might leak out of a plastic bag and into the drug fluid. By forcing all suppliers to standardize this baseline safety data, the biopharma companies are trying to make it significantly easier and cheaper to swap out a basic buffer bag from a premium supplier like Sartorius for a cheaper bag from a competitor.
Without having to redo all the expensive validation testing from scratch.
Exactly. They are actively trying to commoditize the easy stuff to claw back some margin.
So logic dictates that the true economic profit, the massive defensible margins, must be migrating downstream.
They are moving to the most complex, highly engineered, difficult to replicate filtration and chromatography steps.
The migration of the profit pool is driven entirely by the asymmetry of risk. I love that phrase from the sources. Let's break down that asymmetry.
Okay.
If a basic upstream media bag fails on day one of a batch, say it leaks or the nutrients are slightly off in, you lose a few days of time and some raw materials.
It's frustrating, and it costs money, but it's early in the manufacturing process. The financial hit is contained.
Conversely, if a highly specialized virus filter fails at the very end of the downstream purification process, the consequences are catastrophic.
You have just contaminated and completely destroyed a fully processed, highly concentrated batch of a commercial drug.
And that single batch could be worth $50 million on the market.
And that is why downstream processing is immune to the price pressure we see upstream. A biopharma company is not going to risk a $50 million batch of a blockbuster drug just to save 10% on a generic, untested virus filter from a new entrant.
The mechanism protecting the downstream profit pool is pure risk aversion, heavily enforced by regulatory oversight.
The risk profile heavily favors established, premium incumbents with flawless quality track records and deep regulatory validation.
Knowing that the real economic value is trapped in complex downstream filtration begs a massive question. How does a company like Sartorius legally and structurally guarantee that a pharmaceutical giant chooses their filter over a competitor's filter years before the drug is even approved?
Because they aren't just sitting back and hoping their reputation wins the contract.
Right. They have a highly aggressive, deeply integrated strategy to secure that lock-in.
The strategy is brilliant in its foresight. Sartorius operates as what they call a total solutions provider. They want their equipment touching every single step of the manufacturing process.
But their ultimate strategic weapon is how they acquire customers at the absolute beginning of the drug development journey.
We are talking about the Amber 15 and Amber 250 systems.
This is the ultimate Trojan horse strategy. My understanding from the technical specs in the sources is that these are highly automated, miniaturized bioreactor systems. They're used in the preclinical process development phase.
We're talking incredibly small volumes, 15 milliliters or 250 milliliters.
15 milliliters is what, like a tiny shot glass?
Basically, yeah. To visualize this, it is a robotic testing station where process development scientists can test dozens or even hundreds of different cell lines, media compositions, and environmental conditions simultaneously.
They are searching for the absolute optimal recipe for manufacturing the drug.
But how does selling a tiny 15 milliliter robotic tester secure a massive 10-year consumable revenue stream for 2,000-liter commercial bags down the line?
It leverages human behavior, specifically the engineering principle of translating scale. The Ambry system utilizes proprietary Sartorius software, Sartorius sensor algorithms, and Sartorius digital twins.
If a process development scientist spends 2 years meticulously optimizing a fragile biological process on an Amber system, that process becomes uniquely tuned to Sartorius's specific digital and physical architecture.
So when that drug finally shows promise and the company needs to scale up for phase I clinical trials, the scientist is going to look at the engineering team and say, do not change a single variable. We know it works perfectly on the Sartorius algorithms by the larger Sartorius pilot-scale bioreactor.
Exactly. The process naturally transfers from the 15 milliliter amber scale to a 50-liter pilot scale and eventually to a 2,000-liter commercial scale, all staying within the Sartorius ecosystem.
The amber system acts as a sophisticated wedge.
It secures an early lock-in at the R&D stage, structurally guaranteeing that when the drug finally achieves FDA approval, the massive, highly profitable commercial-scale single-use bags and downstream filters will also be Sartorius products.
It's an incredible mechanism for future value capture. But as dominant as that strategy is, Sartorius is still fighting in a fierce oligopoly. We need to assess the competitive landscape, looking at where advantages are expanding and contracting.
Let's start with the incumbents. Danaher and Merck KGaA. Where is Sartorius vulnerable against them?
The Unfiltered Expert Network calls highlight a very specific structural vulnerability for Sartorius at the enterprise level. Danaher and Merck KGaA are massive, diversified conglomerates.
They possess a much broader enterprise portfolio than Sartorius.
Right. They aren't just selling bioprocessing equipment.
Precisely. This vast portfolio allows Danaher and Merck to execute massive, enterprise-wide corporate agreements. They can approach the C-suite of a global pharmaceutical giant and offer to bundle all of their bioprocessing equipment with their basic laboratory chemicals, their diagnostic research tools, their mass spectrometers, and their clinical software.
They can offer massive top-level negotiating discounts across a huge suite of completely unrelated products to win the bioprocessing contract.
The expert transcripts noted that this gives Danaher and Merck a top-level negotiating advantage.
Because Sartorius, being more of a pure-play bioprocessing and lab essentials company, lacks that massive enterprise leverage in C-suite procurement discussions.
They have to win purely on the technical merits of the bioprocessing equipment itself.
Okay, so that is the threat from the massive conglomerates above them. But what about the threat from below? The challengers aggressively trying to innovate their way into the profit pool. The sources heavily feature a mid-sized competitor called Repligen.
Repligen represents a highly potent, targeted threat. They are deeply focused on bioprocessing, much like Sartorius, but their competitive advantage is expanding through aggressive technological integration.
Specifically, they are leading the charge in integrating what the industry calls process analytical technology, or PAT.
Let's clearly define the mechanics of PAT, because it seems critical to the future of the industry. Historically, if you wanted to test the quality of the drug inside the bioreactor, you had to physically draw a sample out of the vat, walk it over to a separate analytical lab, run a test, and wait hours or days for the results.
It is a massive bottleneck. It introduces the risk of contamination every time you take a sample, and it prevents real-time adjustments to the batch.
But PAT changes that entirely. My understanding from the sources is that PAT involves engineering advanced, miniaturized sensors directly into the manufacturing line.
You are testing the active drug fluid in real time, at line or in line, completely eliminating the separate lab.
And the sources highlight Repligen's recent acquisition of a company called 908 Devices.
That's a huge move.
It is. This acquisition specifically brings advanced at-line and online mass spectrometry testing capabilities directly into Repligen's portfolio.
By offering smarter, more deeply integrated real-time analytics, Repligen is aggressively threatening to capture a significant portion of that high-margin downstream value that Sartorius relies on.
And then we have the new entrants, the substitution risk. And this is where we have to apply absolute intellectual honesty and look at the brutal reality of the Asian market.
The vulnerability here is profound. If you look strictly at Sartorius's investor presentations, management boasts about their premium market positioning, their incredible historical margins, and their technological superiority.
But the raw, unfiltered expert calls paint a much more friction-filled picture.
I found these quotes to be the most revealing part of the entire stack. Independent consultants and former competitors explicitly stated in these interviews that emerging, smaller biotechs increasingly view Sartorius as, quote, too expensive.
That perceived price friction is the exact wedge that emerging Asia competitors are leveraging to steal market share. The domestic biopharmaceutical industry in China is booming, and local suppliers are building massive manufacturing capacity with significant structural cost advantages.
But wait, if the regulatory lock-in is as powerful as we established with the Trainer Inc. analogy, why would a biopharma company risk using an untested Chinese buffer bag just to save a few pennies? Isn't the cost of FDA revalidation too high, even for the cheap stuff?
That is the exact tension in the market right now. For a fully validated commercial blockbuster drug, you are absolutely right. The biopharma company will not switch. The lock-in holds.
So where are the Asian competitors actually winning?
They are winning at the beginning of the pipeline. If you are a small, newly funded biotech startup in a tight venture capital environment
Right.
and you are just beginning preclinical trials, you haven't validated anything with the FDA yet.
You are extremely price sensitive.
When you need a massive volume of basic, low-barrier upstream products, standard cell culture media, simple mixing bags, basic buffers, you might look at Sartorius's premium price tag and choose to validate your early process using the significantly cheaper Asian supplier.
Because the risk of failure in early upstream steps is lower, and the startup needs to stretch their funding.
Exactly. The competitive advantage of the premium incumbents is contracting in the low-barrier commodity space.
The experts explicitly noted that to maintain their historical growth rates in the face of this intense price competition, companies like Sartorius will be forced to fiercely defend and expand their complex downstream technologies.
They cannot win a race to the bottom on price against massive Asian capacity.
This intense competitive pressure naturally forces us to look forward. We've mapped the history, the structural economics, and the current battlefield. But looking at the next 10 years, the fundamental nature of how a biological drug is invented and produced is changing.
We need to analyze who makes the pie bigger, which is value creation, and who actually manages to eat the pie, which is value capture.
Let's begin with value creation. How does the bioprocessing industry physically grow larger over the next decade? The most significant driver of value creation is happening at the very top of the funnel, in early drug discovery.
And the sources point directly to the explosion of AI-driven drug discovery. We are talking about breakthrough computational tools like AlphaFold.
The implications of tools like AlphaFold cannot be overstated.
Huh.
Historically, mapping the 3D structure of a complex protein, which is necessary to understand how a drug will bind to a disease target in the human body, could take years of painstaking laboratory work.
AlphaFold and similar AI models can accurately predict these 3D structures computationally in a matter of days.
One of the former bioprocess marketing heads interviewed in the Expert Network calls noted that AI tools will soon allow pharmaceutical companies to simulate immune system responses and complex molecular binding setups entirely digitally.
By moving the trial and error process from the physical lab bench to the digital simulation, AI effectively carves out entire years of preclinical development time. It drastically lowers the cost of failure in the early stages.
And if you lower the cost of discovery, you drastically accelerate the sheer volume of viable molecules entering the clinical pipeline. Furthermore, it's not just traditional monoclonal antibodies filling this pipeline.
The sources highlight the rapid rise of advanced therapies.
We are seeing a surge in antibody-drug conjugates, or ADCs.
These are highly complex molecules that essentially attach a targeted chemotherapy toxin directly to an antibody, allowing the drug to seek out and destroy cancer cells without ravaging the healthy surrounding tissue.
We are also seeing the continuous advancement of cell and gene therapies, which, despite their current reimbursement hurdles, represent the absolute frontier of personalized curative medicine.
So when you combine AI-accelerated discovery with an explosion of complex new modalities, the sheer volume of drugs needing to be manufactured is going to explode.
This massive influx of pipeline volume is the fundamental mechanism that preserves the historical 10% baseline industry growth rate that the experts keep citing. AI creates massive value by ensuring the pie keeps getting bigger.
But as we analyze the market structure, it is critical to remember that a larger pie does not automatically guarantee that Sartorius captures a larger slice of the economic profits.
That brings us to the fierce battleground of value capture. And for the next decade, that battle revolves almost entirely around one massive, elusive engineering concept: continuous manufacturing.
Let's thoroughly explain the mechanics of continuous manufacturing, because up until now, everything we've discussed involves batch processing.
In batch processing, you fill a large bioreactor bag with media and cells. You let it brew for several weeks, you completely empty the bag, and then you take that harvested fluid and run it through a series of sequential downstream purification steps.
It is an inherently start and stop, fragmented process. Batch processing, even when utilizing highly efficient single-use bags, is fundamentally inefficient from a time and asset utilization perspective.
Continuous manufacturing is the holy grail of bioprocessing.
The fluid dynamics of continuous manufacturing are entirely different. Instead of a start and stop batch, you constantly feed fresh raw materials and nutrients into the bioreactor at one end, and you simultaneously constantly extract and purify the finished drug fluid out the other end.
It is a 247 uninterrupted flow.
It replaces a giant slow cooker with a highly calibrated, continuous fluid conveyor belt.
The economic value capture here is immense. If a company can successfully engineer and patent a true end-to-end continuous manufacturing system, they can offer pharmaceutical customers unmatched cost of goods reductions.
Because you can produce vastly more drug product in a significantly smaller physical footprint over the same period of time.
The sources indicate that Sartorius is investing massive amounts of capital to win this race. They have partnered extensively with Siemens to develop the digital software architecture required to run these systems.
And that digital partnership is the crucial mechanism. You cannot have a human technician manually checking valves and flow rates on a continuous 24/7 biological fluid stream.
It requires intense, flawless automation, predictive analytics, and deeply integrated PID sensors to constantly monitor and adjust the flow in real time.
Whoever owns the software ecosystem that makes continuous manufacturing possible will capture an outsized portion of the industry's profit pool over the next decade.
But this intense drive toward continuous manufacturing and higher-yielding biological clones create a massive contradictory ripple effect. We have to look at the second-order effects of all this process intensification.
If biological titers keep rising, meaning the cells secrete more and more drug per liter, and if continuous manufacturing actually succeeds in shrinking the required physical footprint, the industry faces a fascinating paradox.
The paradox of overcapacity.
Exactly. An expert from Novartis specifically highlighted this threat in the transcripts.
If the global bioprocessing industry can suddenly produce massive amounts of commercial drugs in tiny, highly intensified continuous single-use systems, what happens to all the millions of liters of traditional capacity currently being built by CDMOs globally?
It severely threatens the utilization rates of massive capital investments. We already saw the canary in the coal mine with the highly publicized struggles of CDMOs like Resilience.
They built out massive physical capacity very quickly, assuming endless volume growth, and subsequently had to shutter sites and restructure when the technological efficiency and the capital cycle shifted against them.
If the physical footprint required to manufacture a blockbuster drug shrinks significantly, the sheer number of large-scale 2,000-liter bioreactors and massive consumable bags sold might actually decrease, even if the total number of approved drugs increases.
The volume game changes entirely.
And the second major consequence of this shifts us back to the Asian competitors capturing the low-end commodity market.
If incumbents like Sartorius and Danaher are losing the massive volume game on basic upstream buffers and bags due to intense price pressure and commoditization, they must find new ways to maintain their massive corporate gross margins.
Which forces them into a frantic, highly defensive M&A race. The sources clearly document this trend.
We saw Sartorius aggressively acquire BIA separations to capture their highly specialized proprietary monolith purification technology for gene therapies.
We saw them acquire Zell to capture highly specific viral vector media capabilities.
The strategic imperative is clear. The dominant incumbents will be structurally forced to continually acquire highly specialized, patent-protected, downstream analytical and purification technologies.
Buying that complex intellectual property is the only viable mechanism they have to maintain their pricing power and defend their value capture against low-cost commoditization.
Let's tie this massive web of data, history, and structural economics together, based strictly on the analysis of the inflection points, the profit pool migration, and the expert network intelligence, who wins and who loses the 2030s.
Let's make some definitive predictions, but crucially, let's explicitly articulate the mechanisms by which these companies will win or lose.
Based on the structural evidence, the clear winners of the next decade will be the highly integrated premium incumbents, specifically Sartorius, Danaher, and Thermo Fisher. But their victory will be concentrated almost entirely in complex modalities and downstream processing.
And the fundamental mechanism by which they win is risk aversion. It's that exact asymmetry of risk we dissected earlier.
The economic reality is that biopharma companies will absolutely refuse to risk a $50 million commercial batch of a breakthrough drug just to save 10% on a generic Asian virus filter.
The mechanism is pure, unadulterated fear of failure, heavily enforced by the immense burden of FDA and EMA regulatory validation.
As long as the regulatory penalty for changing a validated process remains sky high, the premium incumbents mathematically win the downstream game.
And looking at the other side of the ledger, the losers are defined by their inability to adapt to the changing physics of the manufacturing floor.
The structural losers will be the traditional, inflexible CDMOs anchored entirely to massive legacy stainless steel infrastructure.
But crucially, the sources also point to mid-tier single-use providers as massive potential losers.
I'm talking about companies that just manufacture the plastic bags but lack a proprietary, deeply integrated software and analytics ecosystem.
The mechanism of losing for these players is structural inflexibility and the hollowing out of the middle market. Legacy CDMOs will be progressively squeezed out because their massive steel tanks cannot offer the nimble, highly flexible footprint required to manufacture highly targeted, low-volume, personalized medicines like autologous cell therapies.
And the mid-tier bag makers will lose because they are caught in a classic margin trap. They cannot compete on pure price at the bottom against the emerging massive-scale Asian challengers.
And simultaneously, they lack the massive R&D budgets to compete on analytical sophistication and software integration against giants like Sartorius and Repligen at the top.
They offer no unique value proposition in an increasingly polarized market.
However, applying absolute intellectual honesty to this deep dive requires us to acknowledge the fragility of these predictions.
We must clearly define the specific invalidation conditions, the scenarios that would completely shatter Sartorius's economic moat and upend our entire analysis.
The most profound invalidation condition is the regulatory wildcard, universal standardization. We established that the moat relies on proprietary validation. Sartorius locks you into their specific bag dimensions and their specific tubing connectors.
But what if the federal regulators change the rules of the game? What if the FDA or the EMA, operating under immense political pressure to drive down the spiraling costs of national health care, suddenly mandate universal engineering standardization for all bioprocessing equipment?
If the regulators decree a 2,000-liter single-use bag must meet these exact universal dimensions and utilize these exact open-source connection standards, regardless of which company manufactures it, the proprietary lock-in vanishes overnight.
Sartorius's premium valuation evaporates instantly under that scenario because its switching costs drop effectively to zero. The printer ink analogy fundamentally breaks down because the government legally forces the printer to accept all ink universally.
While highly unlikely in the near term, given the FDA's current posture on risk, it remains the absolute greatest existential threat to the industry's profit structure.
And the second major invalidation condition surrounds the immense hype of continuous manufacturing. The corporate presentations paint a very clean, triumphant narrative that Sartorius will perfectly execute this transition and capture the ensuing value.
The unfiltered expert sources demand deep skepticism regarding that narrative. The evidence on the actual feasibility and widespread adoption of true continuous manufacturing is genuinely ambiguous.
One highly experienced former executive explicitly called continuous manufacturing a tough nut to crack that the industry has been confidently talking about solving for 20 years.
If continuous manufacturing remains an elusive, capital-intensive pipe dream that never fully scales across the industry, or worse, if a completely disruptive outside software or fluid dynamics startup solves the digital control problem before Sartorius does, the value capture narrative shifts entirely.
We cannot manufacture a clean winner-take-all prediction when the fundamental engineering challenges remain unsolved. The industry structure currently dictates persistent uncertainty regarding how the most advanced modalities will actually be scaled in the 2030s.
The battle between biological complexity and engineering efficiency will define the next decade of capital allocation.
Which leaves us with one final, completely provocative thought to end on. We've spent this entire deep dive analyzing massive, centralized factories. Even if those factories are transitioning from rigid steel to flexible plastic bags, they are still massive, centralized hubs, shipping complex drugs across global supply chains.
But if personalized medicine, specifically autologous cell and gene therapies, becomes truly hyper-individualized,
where a drug is engineered from a single patient's own harvested cells, modified and re-administered in a matter of days.
Will the future of bioprocessing eventually move out of massive centralized factories entirely?
Imagine a world 10 or 20 years from now where manufacturing doesn't happen in a colossal facility in Switzerland or a massive CDMO park in South Korea.
Imagine it happens in a miniaturized, fully automated, incredibly sophisticated Sartorius box sitting right next to the patient's hospital bed.
A completely closed-loop bedside system. It draws the patient's cells, genetically modifies them using onboard AI, continuously purifies the bespoke therapeutic, and administers it back to the patient in one uninterrupted fluid loop.
It would completely disintermediate the global logistics network as we know it, turning a massive, centralized industry into a distributed network of bedside appliances.
So the next time you hear about a $2.1 million miracle cure or a massive breakthrough in AI drug discovery, remember, the true marvel might not just be the complex biology.
The hidden marvel making it all economically and physically possible might just be the highly engineered plastic bag holding it together.
Thank you for joining us on this deep dive. Keep questioning the invisible structures around you and keep exploring.